Okay, let's discuss inversion heterozygotes now. So here's an organism that has the same chromosome that we've been dealing with now with an inversion event that has broken and rejoined this segment of the chromosome. This is an inversion heterozygote because this organism is now heterozygous for two chromosome structures. It's also a trihybrid in the usual sense. It's heterozygous for three loci, as we're used to, but it's also an inversion heterozygote. If this was big A, big B, and big C, we would still call it an inversion heterozygote because of the change in structure on the one chromosome. So let's take this organism and get it ready for meiosis by showing what synapsis would look like for this inversion heterozygote. With inversion heterozygotes, we have a loop, again, like we've seen in deletion heterozygotes, except in this case, both homologs are present in the loop. Again, chromosomes are flexible and able to maximize their pairing. And again, we can put dots out to indicate where synapsis is taking place, and synapsis is taking place within the loop now because both homologs are there. Essentially, what one chromosome is doing is it's twisting and folding up inside the loop of the other to maximize synapsis and maximize pairing. So let's put some loci on here. Okay, one that's in the loop. It's standard practice to show the inverted chromosome as the one that is doing the twisting, although it is rather arbitrary that convention is there. But let's keep with that convention and show that the inverted chromosome is the one that has to twist to match up with the wild type structure on the other homolog. So that would put the dominant loci on the twisted chromosome and little a, little b, and little c on the non-twisted homolog. So here's synapsis ready for meiosis 1. The issue is now, what happens if there is crossing over? Synapsis allows for crossing over, and what we'll do now is we'll discuss what happens when a crossover happens in various locations in this synapse set of homologs. So here's a redrawn image of what we were just looking at with this inversion loop. In this trihybrid. Now, what you can do is start to think about, well, what happens if I have inversions, So here's a redrawn image of what we were just looking at, an inversion loop in this trihybrid. And now it's time to start asking the question, what happens when crossovers happen in various spots within these synapsed homologs? Let's do the first example. Let's put a single crossover inside the inversion loop. And as you're about to see, this is not going to go well. The trick with determining what's happened after a crossover event in these types of structures is to carefully trace homologs based on the centromere that they're attached to. So let's start with this one here. The top homolog here was not involved in the crossover. So we would expect it to have the usual structure and the little a, the little b, and the little c. This second copy here, though, this chromatid, starts off by picking up the little a, then we're into the loop, and then we have a crossover event, and that crossover event puts us down onto this chromatid here. This chromatid, however, is inverted in its orientation relative to the other, but the cell doesn't know that. So as a result, a crossover, when it happens in this region, now puts us on this chromatid, which you can see actually brings us back to the other centromere. So this produces what's known as a dicentric bridge. Dicentric just meaning two centromeres, and a bridge, of course, because it's joining the two of them together. So let's just trace out what we have on this dicentric bridge. We have the little a, then we're up here, we pick up the little b, then we cross over, and we pick up a big A, and then we're back to the centromere. Now, the other one that's down here that is not involved in the crossover has the standard big A, big B, and big C with the inversion on it. And what's not immediately obvious, but if you thought about it and worked it through, there's also an acentric fragment here. This crossover produces something that doesn't have a centromere. We have to trace it from the other side of the equation. So it's this strand here. So it starts off with a big C, then we're up and we pick up a big B, then we undergo the crossover, so we're over onto this chromosome. Right, we're over to, this chromatid right here, and then we pick up the little c, and then we're out to the end without a centromere. So this is an acentric fragment. This acentric fragment isn't going to be inherited by anything because it doesn't have a centromere, so it's going to be lost. And this dicentric bridge is going to break somewhere randomly as the cell attempts to segregate these centromeres by pulling them apart to the poles of the cell. So we're going to have some random break in here. The short answer to what's going on genetically here is every time there is a single crossover within an inversion loop, the products of meiosis that would have resulted from that single crossover will be lost. They won't have a complete set of chromosomes. This acentric fragment will be lost, and this is going to break randomly somewhere, but there's no way to get a full set, a full haploid set of genetic material out of these products of meiosis. So as a result, we're going to see some products of meiosis that fail to survive or abort. So we'll see some percentage of sterility. And the ones that are not involved will produce just the original input types. So what this means genetically, if we go back to this original situation where we were discussing this original inversion event, when we attempt to determine the recombination frequency between A and C, for example, any single crossover inside this inversion loop is going to form a non-functional product of meiosis, or an aborted product of meiosis. So as a result, the observed recombination frequency between A and C, for example, will decrease, because only crossovers that are here, or perhaps out here, can successfully give us genetic recombination between A and C. We haven't worked it through, but it's something that you should do with a piece of paper. Take one of these inversion loops in an inversion heterozygote. Try out a crossover here. You'll find that it doesn't have an impact. It still produces proper haploid sets. We don't have deletions or duplications as a result of an acentric fragment or a dicentric bridge being formed. So crossovers here are okay. Crossovers here are okay. But crossovers within the loop are not okay, in that they produce non-functional products of meiosis. Although I should point out it's single crossover events that produce non-functional products. Double crossover events, in fact, if you work this through, again, you should do this with a piece of paper. Double crossover events, and what we mean by a double crossover event, again, is two single crossovers that affect the same pair of chromatids. You will, if you trace this out, you will find that it restores the proper chromosome structure. So as a result, double crossovers within an inversion loop are fine, but single crossovers are not, because they produce these aberrant products of meiosis. One thing to note is that a single crossover within the inversion loop is what gives us these non-functional products of meiosis. So say, for example, we did a cross and we noticed that 20% of the products of meiosis abort. Fail to complete development. This 20% is actually a detector for single crossover events within the inversion loop. So as a result, we can infer that the inversion loop must be 20 map units long. So this actually tells us physically how large that interval is. So when you're dealing with an inversion loop in a question, typically you'll have some value that tells you how many products of meiosis are aborting, say 20%, so 20 map units. So you have a map that you're dealing with. You have your original map. And you also have this piece of information that there's a 20 map unit inversion somewhere that seems to be affecting these loci because the new recombinant frequencies that you're observing in this cross are different from what you would expect. We have altered recombination frequencies for loci known to be linked on the same chromosome. So you've got your original map, say, from an earlier part of the question. You have this piece of data that there's a 20 map unit inversion, and it's an attempt to place where this 20 map unit inversion is on the chromosome. Recombination between A and B, the only possible recombination space for A and B is this interval. So if you map from A to B in this inversion heterozygote, that A to B apparent recombination frequency will give you this distance from the A locus to the first breakpoint. The recombination frequency between B and C, the only place for recombination single crossovers that's possible would be this particular region here. So the observed recombination frequency between B and C would be this interval here. And then A to C would be the sum of these two distances put together. So what are the diagnostics, for determining that you're dealing with an inversion? Just get some space here. We see a reduction in recombination frequency for known linked loci. And we also see some products of meiosis abort or fail to complete development. This can range from 0 up to 50%. And that's simply based on how large the inversion loop is. Obviously, if it's 0, you won't notice it because it's such a small inversion that it's not affecting what you've previously mapped. But a very, very small inversion will give a small number. So say like a 2 map unit inversion would show that there's 2% products of meiosis that fail to complete development. And this can range all the way up to a really large inversion that would be 50 map units worth of DNA. And a single crossover in that region would produce non-functional products of meiosis. So you could end up ranging up to 50% products of meiosis aborting. So in terms of sterility, which is just another way of saying products of meiosis that don't work, the sterility of an inversion heterozygote will range from 0 to 50%. We will also see a reduction in recombination frequency for known linked loci, perhaps. And these things are indicative of an inversion heterozygote. You distinguish this from a deletion heterozygote because of this. Deletion heterozygotes don't have sterility issues, but inversion heterozygotes do.